So now let's dive into some of that prokaryotic diversity. So we already may have talked about there are two domains of prokaryotes, bacteria and archaea. Here let's talk about what some of the differences are between bacteria and archaea. So looking at this phylogenetic tree, we see our three main domains of life: bacteria, archaea, and eukarya, right? We already know that the very first cell ever produced, ever existed, is represented here by this bottom node right here, the ancestor of all of life, what we call LUCA, last universal common ancestor. And that this node produced two lineages. The organisms that were most like the first cell are called bacteria. So we think that bacteria are probably most similar to what LUCA would have been like. They're very simple, pretty single-celled organisms, but they are super diverse, right? This other lineage over here split again to form two domains. One of those domains is archaea, which is still prokaryotic cells, but the other domain is eukarya. We'll talk more about eukarya next week, but now let's take a look at these two prokaryotic domains right here, bacteria and archaea. So these are all of the clades of bacteria. So that domain of bacteria, that lineage that split into 5 main domains. They exhibit all of the metabolic processes that we mentioned earlier, and sometimes they might have more than one of those metabolic processes. They could present multiple types of those metabolisms. Some are anaerobic, meaning that they don't like oxygen. Some are aerobic, meaning that they need oxygen. Some are nitrogen fixers, some are not. They live in a wide variety of places, deep in Earth's crust, to hydrothermal vents, to frozen in the Arctic. Some of these bacteria cause disease. Some can live in soil. Some live on us without causing disease. Some help us make wine and cheese and also yummy things like yogurt, right? But the key thing here is that they're everywhere. Really. I'm not too concerned that you memorize the 5 names of these clades. That doesn't mean that you won't see this again and we won't talk about it again. But there are a couple things that I do want you to focus on. First, the clade on the far right, Gram-positive bacteria. What does that tell you about their structure? What does that tell you about the evolution of the cell wall? And second, I want you to notice that the sister taxa of Gram-positive bacteria are the Cyanobacteria, those that form stromatolites and do a lot of nitrogen fixation. What does their close relationship with Gram-positive bacteria tell us? Just some things to think about. We aren't going to dive too deep into domain archaea, but there are a few important things that we should talk about. First, we want to know that archaea are prokaryotes. They exhibit a lot of the same features as other prokaryotes. They are single-celled with a pretty simple structure, and they have circular chromosomes. If we look at them with a microscope, they will look like other bacteria. They'll just look like other prokaryotes. We won't be able to tell them apart. There are some pretty significant differences, though, between archaeans and other prokaryotes, especially bacteria. The first is that bacteria have a cell wall made out of peptidoglycan. Archaeans have the cell wall, but it's made out of different stuff. So we don't find a cell wall made out of peptidoglycan in archaeans. Secondly, antibiotics can kill bacteria, but they don't affect archaeans. So that's not a problem though, because archaeans aren't disease-causing, at least not any that we know of. The other main difference between archaea and bacteria is a bit in the weeds for us right now, and it's summarized in your book if you're interested in the specific details, but we haven't covered a lot of those topics yet. So when you look at the table, you might notice that it compares eukarya and archaea and eukarya and bacteria, and that archaea and eukarya share a lot of similarities. And indeed, it's this similarity between eukarya and archaea that supports our current 3-domain hypothesis, as well as how that tree diverged, the branching pattern of the 3 domains of life. Archaea were first discovered in extreme environments. They were first discovered actually not too long ago, and they were originally dubbed extremophiles because they had only been found in really weird or extreme places, like really, really salty lakes, like that pink film that's forming right there in that salty lakes. We found them in hydrothermal vents, or in hot springs, like in that pool in Yellowstone that you see right there in the middle. So we thought first that they really only lived in extreme environments, but we started to find them in other places that weren't as extreme. So archaeans are pretty common in our intestinal tract, and in fact they're pretty common in the intestinal tract of most mammals, especially mammals that eat plants. And they help in break down components of plants that our cells cannot break down. And as a result, of that breaking down of plant cells, they produce methane gas, which we get rid of as flatulence. Cows then burp up this methane gas, right? So that's a byproduct of archaeans that live in our digestive system that help us break down some components of plant cells that our cells cannot break down. But archaeans are also some of the only things that we know of that can live right on some of the hottest deep sea hydrothermal vents, like we see on the lower left here. So what did we learn about how life originated and the role of hydrothermal vents in the origins of life on our planet? And how might that information about archaeans living on deep sea hydrothermal vents help us interpret that phylogeny of life with those 3 domains?